So I just want to tell you about Rady Children's Hospital a little bit. We're the only hospital in San Diego County, Riverside and Imperial counties. We're the largest hospital on the left coast. We have the only level 4 NICU in the county. There's one at the UCSD hospital, but we run that too. We provide care to a little over 250,000 kids a year. The vast majority of those are outpatient. Our ethnic makeup is pretty much matches what we have in the county. It's 50% white Hispanic, 40% white non-Hispanic, and 10% others, with a fairly sizable Pacific Islander population. If kids get sick in Saipan or Guam, they're usually shipped over to us. And we also have a lot of, there's a big Navy base there, so we have a lot of military kids as well. The age of these kids, we'll sequence kids that are about an hour old, and we can sequence, well, not kids, they're adults. So if you get a disease that manifests before you're 18 years old, you can still be seen in a pediatric hospital. However, the vast majority of the kids we sequence are less than 3 months. And what do we get for diseases? Well, we get pretty much everything. We get immune diseases, we get congenital anomalies, but the vast, vast majority are neurological disorders. However, only 35% of our kids get a diagnosis, and this makes me very mad, because that means 65% of our kids are going undiagnosed. We know some small percentage of those probably aren't genetic, probably about 5 to 10%. We've done sort of some long-term follow-up. But the rest of them, it's almost certainly genetic. So what's one of the ways we can get at these kids? Well, one of the ways is long reads, right? And what are long reads good for? Well, you've been here for a day, you know all of this. So dead zones, places where you can't align short reads uniquely. Structural variants. Phasing is actually very important. And methylation, which I'll talk about at the end. This is something we published a little while ago, and it was how to select patients for long read. So if you don't want to sequence all your kids on long read and you want to pick a subset, how can you select them? Well, one of the things that seemed most obvious to us is that we could select based off of phenotype for genes that were enriched for dead zones. So you take every possible phenotype in HPO that is associated with a gene, you figure out which one of those genes have dead zones in them and how big that dead zone is, and then you can figure out which kind of phenotypes are most likely going to help your yield with long reads if you're looking for mutations in these dead zones. And we had a nice talk yesterday that was talking about male infertility, and you can see the vast majority of these, all these things in green, are associated with male infertility. That's because you have this pseudo-autosomal region on the X and Y that make things really difficult. But there's a huge chunk of things that are all immune-related. And some of these seem non-specific, like seizure. But when you look at the underlying genes, they're all immune-related seizure disorders. And so we actually had a particular interest in looking at immune cases. So here's our cohort. We had 55 probands. 52 of them were pediatric, 3 were adults. 11 had known molecular findings, and almost all of them had short-read whole genome sequencing. So just to make things clear, we do not do exome sequencing. All we do is short-read whole genome sequencing and whole genome long-read sequencing. 11 had known molecular findings. These are our positive controls. And 45 had no diagnosis. 25 of them were unselected, essentially. They're consecutive samples from another study that we were doing in the NICU where they just did not have a diagnosis from short read. And 20 were physician-selected samples. So these are samples where the physician is really confident that the kid is genetic, either because of the severity of their disease, or they have a sibling with the same disease, or there's some other kind of transmission. In our diagnosed cohort, we had structural variants, phasing, methylation, and repeat expansions. So repeat expansions, we've gone into this. The disease severity is reported as linked to length. So typically, you'll have sort of a normal length that is quite short, and then you'll get into what they call the premutation, where it's a little bit longer. And then usually, if you're premutation, you'll start having kids that have much longer. And you'll actually see something they call anticipation, which is where, you know, granddad gets it when he's 80, dad gets it when he's 60, kiddo gets it when he's 30. And he typically will not have kids. If it gets very long, you can actually see it in the infant period. It's very difficult for short reads to resolve these. You usually need specialized software, you need locus-specific training, and you may not be able to differentiate between these two different lengths. In pediatrics, we don't care about a lot of these because they usually have adult onset. So I'm a PhD, but I work in a pediatric hospital. You turn 18, I no longer care about you. All right? So there are some exceptions. Fragile X, which is supposed to be about 1 in 7,000. I've never seen one. A bunch of my good neurologist friends have also never seen any. FOX2B, which we see quite a few of. And like I said, sometimes you can get these ataxias in the infants if it's really, really, really long, but it's quite rare. And then DMPK, which is also about 1 in 7,000. Weirdly enough, we have more DMPKs than any other gene at the Institute. I don't know why. So for DMPK, if you're under 50 repeats, you're normal. If you're a bit longer, you're in mild, so you'll have an adult onset, possibly no onset. If you're in sort of the classic, you'll have an adult onset with a slightly reduced lifespan. And if you have quite a long repeat, you'll have congenital onset with a much reduced lifespan. So this is a patient, 8 days old, had encephalopathy, a coagulopathy, large gestational age, premature, dysmorphic, lung hypoplasia. It was undiagnosed, but it did have a novel GUS, a rather large chromosomal duplication. But it also came with us with a known repeat expansion in DMPK. So we did short reads on this kiddo, and so with Dragon Expansion Hunter, we called 128 repeats, which is about 390 base pairs. This would be equivalent to a mild disease with normal life expectancy. But if you're quite savvy, you know this is about the longest that you can call from a short read sequencing. So you'll not see a number bigger than this. So basically, the short read sequencing is only telling us that this is expanded. We don't really know how expanded it is. When we did long read on it and ran Straggler, we got 1,878 repeats. So quite a bit longer. Sniffles also calls it as a large insertion with only a 2% error between the two. So that was kind of nice. And this is congenital disease. So this kid does not have a very good life expectancy. However, this kid also had a southern blot. This is the gold standard. This is the CAP-CLIA. If you have a repeat expansion, we should be running southern blots on it. That calls only 1,326 repeats. That is a 33% difference. That's an interesting thing. I'm not going to answer it for you because I want questions at the end. So I'm going to go on to another case. All right? So this is a molecularly and socially complex case. So this is interesting because fundamentally, when you're working in a hospital system, you have to deal with the most horrible thing in the world, and that is people. So this poor kid, his father is overseas, totally uninvolved. Mother died when the kid was 6. And we often get these situations where the parents just aren't available. And this means you can't phase things. So in normal situations, you can imagine you have both parents. You can very easily phase variants, very cheaply and inexpensively. But if you don't have them, you can't do any of this. Right? So this is why phasing is really, really important to us. So this is actually, I don't know how well you can read that. This is their actual report. Okay? So they had a fairly large duplication. And then they had a number of VUS variants and Ps and LP variants, 2 in UBE3B and 2 in HERC1. And so they asked us to find map this duplication as well as to do the phasing. So what you can see here, so this is one end of the duplication. The blue lines are sort of the blue arrows are sort of showing you where we see the coverage gains. It's sort of indicating the duplication. I'm sorry if you don't look at IGV screenshots all day, but you're very lucky you don't have to. And then the orange arrows, what they're showing us is that they're showing us allelic imbalance. So what we're seeing is that instead of seeing 50-50 of both alleles, we're seeing much more of one of the alleles and much less of the other. So that's another way to indicate that we can actually see this duplication. So this is the phasing across HERC2 over 30 kb. These variants are in cis. So unlikely to contribute to the phenotype of this child. And then we did the same phasing over UBE3B, and these are in trans. And I don't know how, maybe that's not easy for you to see. So you can kind of see that there's 2 groups here. There's a group at the top and a group at the bottom. These are the 2 haplotypes. So you can see the blue arrow at the top there showing one haplotype, and the blue arrow at the bottom there showing the other haplotype. This indicates that they're in cis. So just as a quick review, this kid had a partial trisomy of chromosome 18, 2 VUS and UBE3B, an LP and a VUS in HERC2. UBE3B causes an autosomal recessive disease, and the variants are in trans, so could contribute to the child's eye phenotype. HERC2 causes a intellectual development disorder. This variants are in cis, though, so do not contribute to his phenotype. However, mutations in this gene actually are associated with cancer, and so this kid actually had a liver cancer as part of his condition, and so it might have contributed to that. So this next one is going to be the best structural variant you'll see this month. This child has a very complex cardiac phenotype. I'm not going to read out all those terms for you. All those terms are about how messed up his heart is. He has a bunch of additional phenotypes as well. So this kid is very, very ill. All right? And what we found here was a deletion, another deletion, and a duplication all on chromosome 9, all at the extreme of chromosome 9. And when you see things like that, that's a little hint that those are actually indicative of something else that's going on. This kid almost certainly has a translocation. Translocations are extremely destructive to the DNA, and the DNA, while it tries to repair the translocation, creates these deletions and duplications. We had a cytogenetics report. So this kid actually has a pattern of malformation that is consistent with ring chromosome 9. So he's missing the telomeric regions of chromosome 9. The cytogenetics told us that a lot of the cells have ring 9, but some have monosomy 9, so the ring is lost, and some have trisomy 9, because you have some rings that can't separate. And there's a distribution of these in the kid. So what does this actually look like? So I don't know if there's any microbiologists in the audience, but I hope they're really happy that we're seeing we're getting away from stupid linear chromosomes back to sensible circular chromosomes. All right, so we have a nice circular chromosome there, and you can see the karyotype, the 2 circles there. So you can see, one circle is showing us the ring chromosome, and then you can see it also could be lost. And this was interesting because I was not sure what the structural variant callers would call for this, because it's an unusual thing. You don't see this a lot. And Sniffles did a pretty good job, actually, called that first deletion, and then called everything else as a whole chromosomal inversion. And it's interesting because early structural variant callers would often call whole chromosomal inversions, and they're always garbage. Like, it was always a false positive. But these long reads, instead of giving us these false positives, this is a true positive. Essentially, it's a whole chromosomal inversion. That's what it looks like. But really what it's referring to is referring to this ring structure. So it actually did a very good job. I was really impressed. The CNV caller also did a very good job, essentially replicated what we got from short reads with a few very minor differences. So now I'm going to move on to DNA methylation. You all know what DNA methylation is, and I'm running out of time, so I'm just going to skip this. But I'm going to talk a little bit about imprinting. All right, so the most important DNA methylation in the world is on chromosome X. Right? You have to turn off one of those chromosomes. But there's also, and you can actually, so why do I have a picture of a kitty cat up there? Well, calico cat coats are driven by X inactivation. So wherever you see orange, that's one of the chromosomes being inactivated. Wherever you see black, that's another one. So you can see it's random but splotchy. There's this system called imprinting. So imprinting is where the DNA is methylated depending on the parent of origin. So for a particular gene, it might be always methylated if it came from your dad, or it might always be methylated if it came from your mom. And this is kind of interesting because if you think about it, it can lead to expression of previously silent variants. Right? You can imagine you have a pathogenic variant in the dad, but it's methylated, and passes it on from son to son to son and stays methylated. That dad passes it on to his daughter, then that daughter, if she gets it, every kid she has a 50% chance of getting that pathogenic mutation. So it looks like there's suddenly a dominant disease in this family. There's also the concept of uniparental disomy. So this is where you get both your chromosomes, let's say chromosome 6, from one parent. And this can happen in 2 different ways. You can either get the same chromosome twice from that parent, so you can get mom 6A twice, or you can get mom 6A and 6B, right? So you basically get both from mom. And it's really interesting because in theory, those chromosome pairs, if you get an A and B from your mom, are perfectly fine. Your mom was a baby, grew up, had children, her genes are fine. So how can this ever cause problems? Well, it can cause problems with imprinting, because either those genes will both be methylated or both unmethylated because they both came from mom. So this is another kid who has a family history of Russel-Silversyndrome. So it has scoliosis, tremors, and speech delays. And again, I, for some reason, always put the sick person on the top. So there's 2 people here. At the bottom is a normal person, okay? And there's 2 haplotypes. And what you can see is that you can see this sort of these lines, these sort of histograms of red and blue. And blue is unmethylated and red is methylated. And you can see in the bottom kid, it's about 50-50, right? Half the reads are showing methylation, half aren't. And at the top, you can see it's almost 100% methylated. So that's how we know this guy. And this is a partial maternal UPD of chromosome 7. So that's why he has it. So all the DNA in this particular region is coming from mom. So that's nice, right? If we know exactly where to look, we can detect the methylation. So how can we look across the entire genome without knowing where we need to look to see if we can see dysregulated methylation? This is another locus associated with Angelman syndrome or Prader-Willi. And again, this is another way to look at methylation. Each one of those bars indicates our confidence in a methylation call, and they come paired. And I've tried to indicate that with a little squiggle brackets at the side. And if we start at the bottom, what you can see is that you can see a methylation and then unmethylated. and then unmethylated and methylated. But all pairs, one's methylated and one's not, except at the very top, where both alleles are methylated. So how can we detect it? Well, we can go in and we can take regions. And these could be 1 kb tiles. These could be CPG islands. It could be whatever you want to define. You can just define it in a bed file. And you calculate the minimum and the maximum, so depending on which haplotype. And I've made up these numbers. So I'm saying on the minimum haplotype, the average is 1. We see 1 methylated CPG with a standard deviation of 3. And in the maximum haplotype, for the one with the most methylation, we'll see 45 positions, maybe with a standard deviation of 5. And once we have this data, we can then compare it to new samples that come in. And we see this sample that came in has a max of 45, but has a min of 40. Right? So it very significantly has more methylation on both its haplotypes than the background controls. And so we can actually pull these out very easily. And you can basically come up with z-scores with them. You can plot the z-scores. You can set your z-score limit. You want plus or minus 3, plus or minus 4, whatever you want. And you can just pull out the regions that you're interested in. So when we do this across the cohort, this is what we find. It's kind of a wild range. We have some people with almost no regions that have dysregulated methylation, and a number that actually seem to have pretty high dysregulated methylation. The highest, interestingly, was a Down syndrome child. It was actually reasonably well known in the Down syndrome research community, but actually not very well known in the clinical community. The second highest is a kid with Russel-Silversyndrome, right? So this is one of the kids that I showed you before. They have UPD, so they have a bunch of weird methylation. But then for some reason, we have this adult, a 49-year-old, with a mitochondrial disorder. Very high dysregulation and methylation. And then we have a 4-year-old with tetralogy of Fallot. This is just a number of heart malformations, right? And we don't know why that we see this dysregulated methylation. And it's one of the problems when you get into functional biology, that you don't know if you're seeing the cause or the effect, right? So we don't know if this is causing it or whether this is a sort of side effect from the kid having tetralogy of Fallot, somehow doing something to the DNA in his blood. And we can see we actually have another RSS that looks almost normal. And then just because some people think that maybe we're seeing this increased methylation because the person is 49, well, we also have a 33-year-old who looks more or less normal in terms of methylation. So we don't think those things are driving it. Now, the last thing that we wanted to do was that we wanted to look for novel imprinted regions, because those are really interesting places to find new disease genes. And we did this in a couple of different ways. So we have 4 data sets here. So in the top left, we have a data set that we produced from whole blood using PacBio. In the top right, it's whole blood that we did with ONT. In the bottom right, this is ONT. This was published a couple of years ago. This was done off of cell lines. And in the bottom left, I believe this is whole blood, but it is bisulfite sequencing, whole genome sequencing. And what you can see that's really interesting is that there's things that are specific to each one of the modalities, but there's this huge group in this middle, these 51 genes, where we have 2 different sequencing modalities, and from three different data sets that say these should be imprinted genes that are not seen at all in the bisulfite whole genome sequencing. And it really makes me worry that we're actually missing a ton of imprinted genes. And so we need to really continue on with this work to find all of them and then confirm it. The other thing I don't know if the word problematic is quite right. These are very sensitive as well. So you can actually tune it in to be very, very sensitive to even slight differences in methylation. And that said, however, it's consistent across all our samples. So we're quite confident in these calls. All right, so for conclusions, long-read sequencing is great for dead zones. I think it's the only rational way to select patients for long-read sequencing. Works great for structural variant detection, obviously repeat expansions. They significantly differ from the gold standard. We don't know what's correct. This was a problem that we had when Illumina sequencing was really taking off, sort of 2009. Sanger sequencing has errors in it. We know that. And so we were starting to get to the point where we were starting to believe Illumina sequencing more than Sanger. So when those started to conflict. So the question is this better than our gold standard? Right? And it's going to take a lot of work to see if this actually is better than the current gold standard. And I hope it is, because doing Southerns is terrible. We can detect methylation that's consistent with imprinting defects. And we've done this with a number of samples across a number of kids. And it's pretty obvious. It's a very, very strong signal. And we can use long-read sequencing to replicate known imprinted regions, differentially methylated regions. And we can find unique regions that are shared by other long-read sequencing platforms. But the source of DNA is important. Like, we see big differences between whole blood and cell lines, which is maybe not that surprising. Acknowledgements, all this work, the vast majority of this work was done by Sergey. He's great. And then some of the follow-up work was also done by Prashant. We've got a lot of support from ONT from this. So Andrew Markham helped, Brian, Sean as well. At RCIGM, Wendy sort of helps make these things happen. Luca runs our lab. She produces the best DNA. And so it's just been absolutely fantastic.